today I will speak about whole genome sequencing and how this can be useful to characterize your strain in a culture collection, because this is anyway the topic of this course. And I put some questions for you in the talk just to keep you awake, because it's early morning and as Laura said, because I didn't. Okay. Okay. So mainly the talk is about different examples, how whole genome sequencing was used for different cases and what you can gain from that for the characterization of your strain. Some examples are from here and some examples are from totally different people. Just very quickly, only the first two slides are technical. Oh.
Put it low. Put it small. Put it small. No, put it small.
Okay. So yes, only the first two slides are a bit technical on what is whole genome sequencing. The other things are kind of light examples of where it's used. So whole genome sequencing, I think most of you know what it is. It's basically you take your organism, you extract the DNA, you fragment the DNA in small pieces, and then you sequence them. Then you somehow take all these different pieces and you try to put them together again to get out the genome. This doesn't have to be that you put the whole genome together in one, but you try to make large quantities of pieces that are together such that you can read what is written on the DNA. It's actually pretty simple. And there are two, the two main differences are you sequence something where you already have a reference genome. So it means that if I now sequence another Escherichia coli strain, considering that already so many Escherichia coli strains have been sequenced, I can use an Escherichia coli strain that was sequenced by someone else as the backbone, and what I sequenced, I try and assemble it to this backbone. Like as if you make a puzzle, but you already have a picture of how the puzzle has to be, then it's much easier to make the puzzle than if you have to do it without a picture. And that would be like if you make a de novo sequencing completely of a completely unknown organism. And the difference, so technically there are two different kind of sequencing that you can do. You can do short read sequencing that mainly in real life is Illumina sequencing, or long read sequencing that is PacBio or Oxford Nanopore sequencing, so mini-ion. And the difference is just the length of the pieces that you get out then for your assembly. So for example, if you have to do a de novo sequencing, so if you have to sequence an organism where that was never sequenced before, where you have nothing similar, it's a very good idea to have some long fragments, because the longer the fragments are, the better you know how they are put together. But if I said before, if you have an E. coli strain where you already know more or less how the genomes are organized, then it's fine to take short read sequences and you assemble them there. Nowadays, many people use a hybrid approach where you do both. You do some in a more shallow sequencing depth, you do long reads, and with more investment in more sequences, you make short reads and then you try to put it together. Okay? And this is just a little technical part on how the sequencing is done. Then, of course, there is a lot of bioinformatics involved, and there it depends very much on what you want to do. So I didn't make anything on the bioinformatics because it really depends on which question you have to your genome. And I will now present to you some questions that you can ask on what it is used for. So the first one, and I think it's also the thing that nearly comes to mind to everyone, is metabolic reconstruction. So I have an organism and I want to know what they do. And here there's an example. Who knows what it is? Any idea? Which metabolism? No? There is ammonia going in, and it is anoxic. It's Anamox. So Anamox is anaerobic ammonia oxidation, and in this case of archaea. And it's a very interesting metabolism because this metabolism was predicted. It was said that this metabolism should exist in nature because thermodynamically it is possible to do this reaction. But for many, many years, these organisms were not found until people really looked for it in wastewater. And I think also Manuela, your old boss, was very much involved in this.
Yeah.
And they really looked for this reaction, and then they found organisms that were able to do this reaction. But they still didn't know what happened inside the cell. They were all just kind of predictions. And only then, when they did the genome of the organism, they found the whole pathways, how they were connected, which kind of molecules were transported in and out, and that there are multiple organisms involved sometimes, so how the different organisms interact in these pathways. And I just read for this talk, I read that it is still not completely elucidated how all these pathways work, despite these reactions were described 20 years ago. So it's actually very interesting how much there is to see on what works inside the cell with whole genome sequencing. Okay, so first we can do metabolic reconstructions, and then we can do a hypothesis on niche of our organism. So we have an organism in culture, and we want to see which niche does it have in the environment. And here I show you this graph that I think you've seen yesterday already, that maybe Christiana showed you. So this is Synechococcus, and it's in the Black Sea, and the Black Sea is very deep and dark. And Christiana, actually with our colleagues from Bulgaria, found that there are Synechococcus, so theoretically autotrophic cells, that are fluorescent and live in the depth of the sea, so in the dark and without. And Christiana, who is really very good in isolation of these bacteria, as I think you saw yesterday, managed to isolate 2 strains from the deep layers of the sea. And so we then, Christiana asked the question, what do they do down there? So what is their role in there? And we then, together with our colleague Pedro, made the genome of these strains. And what he saw was that these are actually pretty normal cyanobacteria. They do everything a normal cyanobacteria does. And in fact, also in culture, they go with light and everything. But the thing that they also found was a pathway of fermentation. So then, based on what we found on the genome of these cyanobacteria, we then went to do experiments. And we grew these strains in the dark without oxygen and did different tests with stable isotopes and then nanosims imaging, and could see that, yes, in fact, these bacteria are able to ferment. So when you keep them in the dark and without light, these cyanobacteria that usually have an autotrophic lifestyle and like to be in the light, they can ferment and they can live without light. And this then we came then up with the whole pathway on suggest of which kind of fermentation they do with this purate and lactate fermentation. And this was thanks to the hypothesis that we made based on the genome of this bacteria. Then another quite interesting use of whole genome sequencing, and maybe also something, or for sure something Mary will speak more, Emanuele will speak more about later, is a secondary method. And it's probably also something that many of you, especially people that work with fungi, are interested in. So let's see if you know this. Which do you think is the metabolite that is made here? It's very famous. No? Any guess? Then I tell you. It's penicillin. So penicillin actually has so as Laura said, I work in antibiotic resistance, usually in the environment, and penicillin is, as many of you or most of you know, probably the first commercially used antibiotic. And it has a very interesting history because it was discovered and used just before the Second World War. And so a lot of problems that came out from injuries during the war could be healed with penicillin, no? Because a lot of the deaths are not actually only because the people got shot, but because the wounds got infected. And you can then disinfect the wounds, but you can also treat them with penicillin, and then they will have less problems with bacterial infections. But at the very beginning, when this penicillin was discovered and it was very much used, and for the soldier, the main problem was that the production was very slow. So it was not that the antibiotic was not known. People knew that it was there, but there was not enough because they couldn't produce it fast enough. And so these are some posters that are from the Second World War during in the US, where people were saying, like, here we build a factory to make penicillin, and they put this out there for the workers. Now, work fast, because if you work fast, then we can make more penicillin, and then we can heal our soldiers. And then this one here was like, with a prude poster, like, you have to be careful not to get syphilis or gonorrhea, because if you get syphilis or gonorrhea, you will get penicillin, but this means you take it away from the soldiers. So there was a lot of this kind of also blaming on prostitutes, that they were taking away, they were spreading this disease and taking away the penicillin from the soldiers. So it's a very complex topic and also socially very interesting. Now, I think many of you know there are very different possibilities. So we can take this gene that we know produces penicillin. You can put it on what here they call cell factories, or fungal cell factories, where you put specific genes under constitutive promoters. You put it in yeast or other fungal cells, and you make them overproduce this molecule. And this way you can get much more out in a very easy way. But of course, at the time, this was not known yet. But this is a very important part also of screening for secondary metabolites in genomes. So people just take genomes, they look for secondary metabolites that are similar to things that are already known. So you can find something that is a bit similar to penicillin. So this could be a potential new antibiotic. No? And this is I think, one of the main uses of whole genome sequencing, especially with fungal strains. Then, of course, there's identification. So it's not always super easy to identify bacteria and also fungi. And here is an example where this was important for us, because I studied the microbiome of animals and also Daphnia. And here we saw once in Lake Maggiore that suddenly Daphnia and also copepods were enriched in E. coli. And E. coli is not a normal bacteria for their microbiome. It's usually not there. We screened some other animals, and we found that sometimes they have E. coli and sometimes not, like, can come and go. But we were really curious if this was part of the microbiome of Daphnia, or if this was part of something that came from outside that somehow survived in Daphnia, because you know that E. coli can come out of the wastewater treatment plants or can be a contaminant when people are bathing. And so we isolated E. coli from Daphnia, because luckily it's quite easy to isolate E. coli. Yeah. Theoretically. True. And we isolated them, and then we characterized them. And here, for example, there was just a new article came out that was showing the diversification of E. coli in all different habitats. And it's a super nice article that shows that E. coli is everywhere: feces, intestine, fresh water, meat, and so on, and that all different types of E. coli can be in all different types of habitats, but they have their genome somehow adapted to the different habitats. So we made the whole genome sequencing of our E. coli, which are these two here. We only made two because at the time it was very expensive. And we saw that actually our E. coli are not related to normal water E. coli, but they are related to E. coli that were isolated from poultry meat, that are also quite close to other stool samples and so on. So in fact, probably the E. coli that we found with Daphnia were somehow a contamination that came in there and not an enrichment of water E. coli. And this kind of making phylogeny with whole genomes gives you an incredibly high resolution, because especially for E. coli, it's very difficult to get this high resolution if you don't go for the whole genome. Then the whole genome was actually not really interesting, just to be honest, because it was just a normal E. coli. Okay, then evolution. And here I would like to speak about the most successful bacterium in the ocean, or one of the most successful bacteriums in the ocean, that is SAR11 or Pelagibacter ubique. Are you familiar with this bacterium? It's a very, very small bacterium that lives in the ocean surface water. It's really the super common. And it was described in 2002 that this bacterium can make up like 30% of the bacterial community in the surface oceans. And just shortly after they described this, they also isolated this bacterium, which was very difficult because it was the first time they used this isolation technique, that is dilution to extinction, where you don't plate, but you keep them in water, because SAR11 cannot grow on a plate, it can only be in liquid. It's a purely pelagic bacterium. And they found this, they cultured this bacterium, and then they made the genome. And this genome told a lot about why this bacterium is so common in nature, because the genome was super small. And when they found it, it was the smallest genome of a free living bacterium that was ever found. So here is the genome size, and here is the number of protein encoding genes. And all of the ones that are down here that are smaller, basically all of the ones are pathogens or symbionts. And pathogens and symbionts often have their genome very much reduced because they take things from the host, so they don't have to produce them. Pelagibacter ubique SAR11 is not a symbiont. As I said, it's poorly pelagic, but it reduced its genome to the minimum. So it is maintained by the minimum possible genes that are there. And this allows it to reproduce quite fast, to propagate quite fast, because it saves especially in nitrogen that is important in the ocean. And it has been many years now of genome sequencing of SAR11, and it's a very interesting topic because they now made these, they have hundreds of genomes of SAR11, and you can really see that they are very conservative. So this is mutations that are synonymous versus non-synonymous mutations. This means that a synonymous mutation will still produce the same amino acid, whereas a non-synonymous mutation will change the amino acid and thereby really change the phenotype or make the protein non-functional. And as you can imagine, SAR11 has such a small genome, so it cannot afford to have a lot of non-synonymous mutations, because this will mean that they will, if a gene is not functional anymore, they will probably die. And so they have this very strong prevalence of synonymous mutations. But the interesting thing is that other bacteria from different groups that live in the same place, they have a totally different strategy. So they manage to be in the same place, but they have large genomes and they accumulate mutations in there. And these are especially bacteria that live in very fluctuating environments, because if you have a lot of fluctuations, then you need to be adaptable. And so this changes then their strategy. So I think it's very interesting that with whole genome sequencing, you can go and find evolutionary strategies on bacteria, and thereby also the evolutionary niche. Okay, and you can also, by whole genome sequencing, get information for future cultivation. So here again is SAR11, and in this case, it's the freshwater SAR11. And it's just a funny story from my life, or from my friend's life, actually, which is this Michaela Sacher, that is was my colleague in Zurich, and she had this idea of isolating these freshwater SAR11. She really wanted them. She found them to be super common in freshwaters, and the marine one was already cultured. So she tried to culture it. She was super tenacious in this. She made hundreds of plates, and she never cultured it. She cultured multiple other bacteria that were very difficult to culture and made really nice publications, but never this one. And then 7 years later, an article came out of someone else that managed to culture this, and she was like, oh no. So, and then she looked at the genome, and she found out that actually in the medium that she made from the beginning, that was mimicking lake water, there was one component that we didn't have in the lab. It was some vitamin, but we just didn't have it. And she thought like, oh, who cares? It's not so important. I will do it anyway. And also she kind of forgot that we didn't have that one over the years. And turns out that these freshwater SAR11, it cannot produce this vitamin. So it's really dependent on this vitamin. And she saw that in the genome and she was like, oh my God, yeah, exactly this compound that I didn't put. And yeah, she added it to the medium and now she has an enormous collection of 1,000 strains. No, it's true. So yeah, so it's also very useful to have the whole genome sequenced in order to get information for future sequencing efforts. Good, another very last, the second to last point, which is something that interests me a lot, which is antibiotic resistance. And antibiotic resistance genes can be, you know that they're naturally occurring, no? As we know that penicillin comes from nature, also antibiotic resistance comes from nature. And not every antibiotic resistance gene is risky in the same way. So if I have an antibiotic resistance gene that is, let's say, on the genome of Staphylococcus, for example, then it will probably not matter. It will just be there on the genome, it will be in the environment, it has its own function there, but it will never be really risky for clinic. The real risk of exchange of environment and clinic in antibiotic resistance is if the gene gets mobilized and somehow goes on plasmids. And if the gene goes on plasmids, then there is a possibility of exchange between bacteria from the environment and bacteria from clinic. This is very, very shortly said what we call the cycle of antibiotic resistance. And so one of the interesting things when we look at antibiotic resistance is to localize the genes. Are they just in the genome? Are they mobilized? Are they related to other mobile genetic elements, or are they on plasmids? And this gives you a very different picture on on how risky this gene actually is. And the very last use of whole genome sequencing is, of course, for metagenomics, because what I said at the very beginning, if you have already a genome sequenced and you sequence a new genome from the same species, then it's much easier than if you have to do it from zero. And of course, the same is true for metagenomics. So if I have a strain in culture and I really like this strain, super interesting to me, and I would like to see if this is also common in other environments, I can, for example, take let's say, a marine strain. So I take my genome, and then you take all the samples from a Tara Ocean or some other large sampling campaign, and you can map the reads from those metagenomes to your genome and see how common your organism is in those samples. So now genomes are really important to map reads from metagenomics and to actually get interesting information from them. Okay. And I think yes, so this is just a summary of some examples on what you can use whole genome sequencing for. And I think you got the idea that it's very versatile. You can use it for many, many different questions, but you have to have a question. Because I think a lot of, it's true, because there is a lot of just genome sequence and then people are just mining it to look for something in there. But yeah, that's quite often quite a waste of time, because a lot of these bacteria, when you sequence them, and probably the same is true for fungi, they're just quite normal. And it's just a bit boring. Okay, thank you very much to Laura for asking me to talk, and for the other organizers also for organizing this nice course. And thanks for listening. And if you have questions, let me know.
Perfect. Thank you, Esther. From home, are there any questions? Whoa.
I have a very stupid question.
No, this question is stupid, as always.
When you showed when you tell this story about your friend, and then you show the paper that you have a very nice figure with all the metabolic rules, and etc. Is that an output from a software that does directly that, or is that someone that did that manually?
Yeah, no. At the time, for sure, there was someone that did that manually. Yeah. Okay. That was manually done. Yeah, I mean that. I don't know exactly how they. I don't know these people, but yeah, that was for sure manually.
Okay.
Maybe now there are softwares. I don't know.
There is biorender.
Yeah, but you like where you can put the pathways and they make you the cell. I don't know.
But they have a basic design, and you can really customize.
Okay,
So,
they didn't do
Of course, the premium account, not the not the free one. But you can get ideas anyway. And of course, to build the pathway, then there are software connected to the genome. Again, also that you have to pay for it. But better to collaborate with somebody who can do it. Then that's the easiest way.
Yeah, but I think that was actually really just manually made, probably in use.
The very beginning, yes.
This one here, I think that at least, I mean, probably the pathways are taken from like cake or something and then imported.
Yes,
But yeah,
exactly.
I have a question.
They also said, and I have one.
Yes.
Sorry. No, go ahead. Yeah. From your experience, because you were explaining the difference between doing a whole genome sequence or doing a de novo sequence, so a species that was never sequenced before. So from your experience, do you think doing only the whole genome is enough? Or because I have heard before that it's usually useful to also have the transcriptomics to then help to make the annotation part?
Yeah,
What is
so
your
I think
experience?
that's a bit different if you work with eukaryotes than with prokaryotes, because if you work with eukaryotes, you have a lot of non-coding parts. So the main thing is that as I think probably most of you know, that in prokaryotes you don't have these intons or these parts that are non-coding, or very little of those. And so if you make the genome, if you make the transcriptome, it's better to make the genome. But yes, for eukaryotes, depending on the organism and depending on the load of non-coding parts you have, it is better to make it also with the transcriptome. Because you would just, for example, in rotifers that I know from Diego, they have so many parts that are non-coding and repeats. And they always make the genome with transcriptomics. And they actually assemble it really from the transcriptomics without even doing the genomics, because then they get only the part that is coding out, because otherwise I think you just have so much junk. And it is also very, very difficult to assemble genomes with a lot of repeats, obviously, because, you never know where it starts and where it ends. And so, that is definitely very useful for eukaryotic genomes to do a transcriptome. Yeah.
Do you have something?
Verena had a question.
Question a bit out of curiosity, because yesterday we talked about culture, axenic, non-axenic. So, I guess we all know the importance of having association and symbiote in your culture. So I was just wondering if it happens to you to work with this metabolism and you have got maybe an idea of something that can be really crucial in such association to have as a function.
No, to me, actually not. I really wish it would, though. It would be very nice. But no, I'm not like the over expert. It's not that I do whole genome sequencing all the time. And a lot of the things we do are actually metagenomics, and then we are much more targeted on specific things. And the genomes that I did, really, honestly, they were very bland. They are just, a lot of them could do everything by themselves and so on. But that also, I think, is a bit related to the isolation techniques that we use. But no, unfortunately, I never discovered anything super cool.
Very good.
Yeah, no, because with Pietro, his study is more about how bacteria protect the algal cell. So it's more about secondary metabolites than about actually metabolic sharing in this, which is also very interesting. Obviously.
Question maybe also related to eukaryotes and prokaryotes, but do you have experience in whole genome amplification from single cell?
No, I don't. But I know many people who did it with like MDA and this kind of multiple displacement amplification, of the cells, but I never did it. So I don't know. I mean, the only thing is that you have to be very clean. Did you do anything?
No, but I want to try it.
Yeah, me
I
too.
don't like it really works, so it's up here.
No, but I think it works. So in the story that I told you of my friend that was not able to isolate this because she didn't have the compound and so on, at a certain point they then did genomes from single cells that they sorted with the flow cytometer and did multiple displacement analysis. I mean, I think that nowadays the technology is very good in doing that, and it's not even so expensive anymore to do it. Then I don't know for eukaryotes if it's probably a bit trickier also. But now they also have single cell transcriptomics, so maybe.
But Isabel, this is your student now that is doing that, no? With the one that talk at the Cavallo, that she put the cells and she make stuff.
Former student, she's now doing her PhD in Germany.
Maybe you can share the contact.
Yeah, maybe I can share the contact. But you want from living cells because they are not using living cells. So for instance, for the transcriptomics, this would not be useful anymore because the cell is dead and probably the RNA is gone. So for the DNA, it would still work, but for RNA, probably not. But they have a new technique where they are doing this to have single spores of fungi. So they have a filter, they have the spores, and they are using the microscope coupled with the laser. So under the microscope, they find the spore of the species they are willing to have the DNA. With the laser, they cut around and they have the single spore into a small tube, and then from there they go for sequencing. And they have had some success on this, but then it really depends, I guess, on the species. And for now, they have used these mostly for DNA barcoding. I know that they want to go for whole genome sequencing. I'm not sure how far they went on that part already.
But maybe you can share.
Yeah.
But this is what this is about. Like someone knows.
Yeah. That's her student. She's doing the PhD. Okay. Yeah.